Wait a couple minutes. Make sure you guys look at the bell work. Have some answers for me. For number one, you got 4 choices. There are 4 macromolecules from the last section. And yes, we are skipping 2.4 for now. We will come back to it. It's about chemical reactions and chemical equations. But it fits better when we talk about photosynthesis and cell respiration in those equations. So I think I said hi to everybody. All right. We will start talking, and then as people come in, they can catch up. So number 1 is an enzyme as a type of what? We studied 4 different molecules: carbohydrates, lipids, proteins, and nucleic acids. So which one is a protein a part of? And an enzyme a part of? Yes, Angelina, good. An enzyme is a protein. It's a type of protein. Good. Activation energy. If we split that up to the word activate and then the word energy, what do you guys think that means? What does it mean to activate something? Oh, Shayla, I just saw your question. We're probably not going back till second semester. Good, Leslie. Start, turn on. Good, Viviana. Yep, to activate something means to start or turn on. So then what if when you throw the word energy in there, what do you think energy has to do with turning something on or starting something? This is one of your vocab words for the week, but what do you guys think? When you put the terms activation energy together, what do you think that means? Not sure? Okay, we'll get to it. Turn on or start is good. And then number 3, what's happening in that picture? What is that? Somebody give me a sentence about what is happening in the picture. It's not a trick question. I just want you guys to describe what you see. So which one's opening the lock, Shayla? The gold or the silver key? Keys in a lock, yeah. What's happening with the gold key? Does the gold key fit? The keys are switching. The gold doesn't fit. So does the lock open with the gold? Nope. It only opens with the key that actually fits. Good. So this is going to be relevant later when we start talking about our lock and key model. All right, so our essential question. Same. What are living things made of? We went through elements: carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. We went through compounds, went through water, and then went through our macromolecules. Then you guys all chose a compound found in your body and described it for the discussion, or you should have last week. Now we're talking about a specific compound found in our body called enzymes. Our body would not be able to function without these. So enzymes are catalysts for chemical reactions in living things. Inside of our bodies, there are tons of chemical reactions going on all the time. And enzymes help those chemical reactions along. Catalysts, all reactions need a certain amount of energy to activate or get started, and this is the activation energy. It's the amount of energy needed to get a chemical reaction started or turned on. And then catalysts will increase the rate or speed of the chemical reaction by decreasing the activation energy. So if you don't need as much energy to get started, the chemical reaction proceeds quicker, happens faster. So if you look at the graph, the blue is a chemical reaction that doesn't have an enzyme. It takes a lot of energy and a lot more time to get started and to process. And the red is another chemical reaction that has an enzyme. It doesn't need as much energy to get started, and it happens faster. Enzymes are catalysts that are found in living things. So chemical reactions happen all the time, but in our bodies, these catalysts are called enzymes. They're needed for almost all processes in our body. Most enzymes are proteins. And then chemical reactions in our bodies that use enzymes include digestion, metabolism, muscle contractions, and bone formation. To describe how enzymes look and how they work and how they fit together, scientists have come up with a model. We call it the lock and key model. An enzyme structure only allows certain things to bind to it. Just like a lock and a key, if you have a key to your house and you go try to use it on your neighbor's house, it's not going to work. Your key will only work on your house. Each enzyme is like a lock. There's only one specific key. That key in scientific terms is called the substrate. And the place where enzyme and substrate come together is called the active site. And a couple different things can happen in a chemical reaction. On the left, you can see the blue is the enzyme, the green is the substrate, and the green splits into two. And those end results are called products. And on the right, you have a different kind of chemical reaction. Wait till it starts over. You have two different substrates that will come together in an enzyme. Those look like puzzle pieces. You can see they fit perfectly. And the chemical reaction works to glue those two substrates together to form one product. So chemical reactions can happen either way. They can split something up or they can glue something together. And enzymes do not change during a chemical reaction. Enzymes don't get used up. Only the substrates change. Like I said, they either break up or get glued together. They're always floating free in our cells to speed up as many reactions as possible, the enzymes are. And then you have a good picture there with the different pieces. You have the enzyme, the substrates, where the active site is at. They come together. And when they get glued together, they're called a product. You don't have to draw this, but it's a good picture to know and to save and to have a reference about. You have this picture on your version of the notes. Disruptions. Enzymes function best within a small range of conditions. All enzymes have optimum conditions that they work best at. Optimum is a synonym for ideal. If you remember the story of Goldilocks and the Three Bears, Goldilocks is a little blonde girl, goes skipping through the woods, comes upon a cabin, barges in, There's 3 bowls of porridge, which is kind of like oatmeal on the counter, and she's hungry, so she sits down to eat them, and one is too hot, one is too cold, and the third one is just right. So that just right conditions is called the optimum conditions. And each enzyme has its own optimum conditions that they like. Temperature is one of these conditions that need to be ideal. pH, most enzymes don't like it too acidic or too basic. Our stomach acid is an exception. Its ideal is acidic. And then ionic conditions, which usually means salt concentration. There's other ions, but salt is a huge one that we use inside our bodies and in nature. There's fish that have to have the right amount of salt concentration to live. If they live in the ocean, they need a lot of salt. If they live in lakes, they need a little bit of salt. And if you switch them, they'll die.